Hello friends, I would like to talk you or discuss about the blueprint of life. What does it mean? Can you tell me? The blueprint of life means the framework on which the life depends, and this is particularly physical frameworks, physical foundation on which the behavior of life, structure and function of life depends, and that is called deoxyribonucleic acid. In short, we say DNA. This phrase is so popular that sometimes you must have heard leaders to say, what is the DNA of this country, what is the DNA of this state. Now, look at the board before us. Here, blueprint of life, DNA. This is the basis of inheritance. DNA, first of all, I would like to talk to you the discovery of DNA. Actually, deoxyribonucleic acid is a sort of nucleic acid. It was first discovered in 1869 by Frederick Nasher in the pus cells of some animals, and since then the chemical constituents of this acid and the structure of this chemical have been started, and now the structure and function both are very clear to all the world's population, what the DNA structure and its function. Then the question, in which place, where this DNA is found. You know, each and every organisms is made up of smallest cells, you know. These cells have nucleus inside them. Nucleus controls and governs whole of the cell physiology and behavior of the cell and whole of. So, here you see, this is the typical cell surrounded by cell wall or cell membrane, and inside the cell there is a nucleus, you see, and inside the nucleus the material you see is known as the chromatin fibers. These chromatin fibers are at a particular cell division state. They condensed and take shapes of chromosomes. It is very well known, the number of chromosomes or constant for a particular species or members. So, this is the chromosome. Here, see, these chromatin fibers ultimately made up of chromosomes. This is one chromosome. Chromosome is the condensed phase of nucleic acid, deoxyribonucleic acid particularly, and proteins, histone proteins. Histone proteins provide the basis upon which the DNA is coiled, highly coiled. So, this DNA is located in chromosomes inside the nucleus. And here, if you reveal the structure of chromosome, you will find this helix type structure, double helix. You see double helix. So DNA is the double helical structure. In this double helix, you see there are two strands, and both these strands are joined together by hydrogen bonds. The black line bars are shown, shows the hydrogen bonds. Triple, double, triple, double. So, in itself, the DNA molecule is double helix. It is highly coiled and it is condensed in chromosome at a particular state of cell division and found inside the nucleus. Now, DNA's structure. Structure means how consists of this strand. Chemically, DNA is a polymer. You see double strand, and these double strand each consist of monomers, and monomers known as nucleotide. Nucleotides. These are the monomers which constitute entire polymers. So one strand is a polymer, means you can say polynucleotides. If each nucleotide combine together to form a long strand, that is called polynucleotide. So two polynucleotides when combined through the hydrogen bonds, then form double strands, and when double strand coil to each other, that is called double helix. Now we concentrate upon the nucleotides. What are nucleotides? Nucleotides are basic building block or monomer to constitute the entire polymer. So nucleotide in itself consists of chemically one pentose sugar molecule. Pentose sugar, you know, it is the saccharide, polysaccharide, and nitrogenous bases. Nitrogenous bases means which include in their structure nitrogen. There are two types of nitrogenous bases groups: pyridine, purines. Here guanine is the purine type of nitrogenous base, and guanine, adenine also another purines. Adenine and guanines are purines. These are called nitrogenous bases. Now another two kinds of nitrogenous bases, cytosine and thymine. These are the heterocyclic compounds. Heterocyclic means in their cycle there are nitrogen atoms and oxygen atoms. So purines and pyrimidines, two groups of nitrogenous bases. First pentose sugar, and then another category is called phosphate. Now when these three triose units come close together to form bond, they constitute nucleotides. Like this, here this is pentose sugar, this is pentose sugar. Attached to this one of the carbon atoms, here purines and another side the phosphate, and it is the carbon 5 is carbon 3, 1, 2, 3, carbon 3. Next and then continues. So now there are bonds, 3 bonds, So this whole unit is called nucleotide. It has a phosphate and pentose sugar and purine bases. So concentrate upon this strand, and the one unit is nucleotide. Here nucleotide, this is the base, this is the pentose sugar, sorry, and here is the purine base, and here phosphate. Now you see the OCH2. Here you see the bond. This bond is called phosphodiester bond. This bond here, this is the phosphodiester bond, because a sugar and a phosphate, when it is combined through this bond, is covalent bond and phosphodiester. You know, base and alcohol when react, here OH group, it forms the esters. So phosphodiester. Now this side, this is purine base bonded another side of sugar, and this is also covalent bond, and this is called glycosidic bond. And now see, there ultimately these bonds are called hydrogen bonds. So a nucleotide is made up of 3 units, and they are bonded by covalent bond, phosphodiester bond on the backbone side of the polynucleotide, glycosidic bond between base, purine base and sugar, and finally hydrogen bond. And when we consider the second strand, this strand continue, and it is just antiparallel of this. So here another side, purine bases, always, as you know, there is a rule of base pairing that was discovered by Chargaff, and this was great hint in finding out the structure of DNA in 1953. Now what is the base pairing rule? Base pairing rule is nothing, but it is these purine bases are pyrimidine. Adenine always combined with the thymine. Guanine always combined with the cytosine. So this is the base pairing. It is fixed, and that's why in any DNA sample the ratio of these pyrimidines are almost equal. So here always guanine attached to the cytosine. Here you see guanine. Look at here, adenine always bonded with thymine, guanine always bonded with the cytosine. Again, adenine thymine bonded by 2 hydrogen bond. Guanine cytosine bonded by 3 hydrogen. So 2 strand means 2 polymeric strand, means 2 polynucleotide strand combined are bonded through hydrogen atoms. So in nutshell, this is the chemical structure of DNA macromolecule. This distance is 20 angstrom almost between 2, and now here the distance between this is 3.34 angstrom, and the distance between 2 strands here is 20 angstrom. Now, so this is all about the chemical structure. This structure, dear friends, this beautiful structure, symmetrical structure of this unique molecule, that is macromolecule of the universe. In another words, there is no match to this molecule. It is made up of For the sake of knowledge, I would like to tell you the human genome is made up of almost 3 billion nucleotides. Consider. And particularly 20 or 25,000 almost genes. They are functional. And most of 89% of human genome is non-functional. So here this polynucleotide structure was discovered in 1953. The well-known scientists James Watson and H.F. Francis Crick in 1953 they revealed the structure. They analyzed the data given by Maurice Wilkins and Rosalind Franklin, who provide the X-ray data, X-ray diffraction pattern molecule, and Watson and Crick. They analyze the data and ultimately using the base pair rule and another properties of the DNA gave out this structure, and they shared Nobel Prize in 1961. So this was discovered in 1953. Again, there were several modifications, different types of polynucleotide. You know, there are two categories basically: RNA, ribonucleic acid, and deoxyribonucleic acid. What is the difference? Deoxyribonucleic acid includes in their structure deoxyribose sugar. Here at the second place of the carbon, there is no oxygen. So deoxy, without oxygen, deoxyribose sugar. And if here will be oxygen, suppose here will be oxygen, so it will be ribose sugar. So the structure of polynucleotide, when ribose sugar included in their polymer, so it is called RNA, ribonucleic. This is mostly single stranded, and DNA is double stranded. So there are two types RNA. Now again, what is the function? This is the structure. Now function is enormous. Function is chemical functions. It behaves as autocatalyst and heterocatalyst, both the function. Autocatalyst, when it acts as an autocatalyst, means it replicates its structure, means it forms copies, and known as replication, DNA replication. It produces own copy with the help of several enzymes, DNA enzymes. So during cell division, prior to cell division in every cells, there is a duplication of DNA molecule. This DNA fiber, this double helix, uncoiled and again copy their original DNA. With several enzymes, participate in this function. Now, heterocatalytic also provides the messenger RNA. It provides template for messenger RNA to be formed. And messenger RNA took the information from the DNA molecule, which is encoded in the form of nucleotides. Messenger RNA reaches to the ribosomes, and with the help of transfer RNAs, this mRNA plays important role in protein synthesis. This process called translation. So, formation of messenger RNA on the any one of the strand of the DNA is known as transcription, and the mRNA, when becomes the base in ribosomes, and with the help of transfer RNA, it encodes, it translates the nucleotide into amino acids. So long chain of amino acids, you know, it is also polymer of amino acids called proteins. So that's why its central function, DNA's central function is replicate itself, also governs the formation of proteins. And one thing more about, not all the DNAs in that cell are functional. Takes part in the formation of proteins. Sum of its parts non-functional, sum of its parts is functional. So polymer, this is a polymer, that is functional. And proteins, you know, general building block of the body constituents proteins, and proteins behaves as acts as enzymes, catalysis, biological catalyst. Almost every function, every biological chemical reaction controlled by the enzymes. Enzyme in turn are made up under the control of messenger RNA, where the information of DNA polymolecule encoded. So in this case, this is called blueprint of life. This way we can say that DNA molecule is the blueprint of life. It governs proteins, and ultimately proteins governs our behavior. So it is the physical basis of our behavior, physical basis of our function or structure. Now most important function is that it takes part in inheritance. Inheritance means the transmission of parental character to the next generation, to the reproduction. So it takes its property, it carries transmitted property DNA from one parent to children, from children to another children. So this is the basis of inheritance. So when Frederick Messer discovered in 1869 the nucleic acid, the function was not known. In 1945 roundabout, the three scientists McCarthy, MacLeod and Abel hinted that its function might be possible in inheritance. So in 1952, Hershey and Chase, in their famous experiment on T-phase, bacteriophage, undoubtedly cleared that it takes part in inheritance. So its function could be known for a long time after the discovery of its nucleic acids. So, and next one, now we use this DNA polymer as a routine basis in chemical laboratories to modify organisms. When we change the DNA, you know, a structured gene, very popular word, gene in biology. So gene, what is gene? Gene is a unit of gene is unit of formation of protein, and this gene constitute ultimately in turn of DNA. So gene a particular sequence of DNA, the nucleotide that governs a simple a particular protein or catalyst, and which, so genes nowadays are chemically synthesized. This polymer no more has a natural polymer, but we synthesize in vitro the DNA polymer, and we introduce and we cut, we splice this DNA in the recombinant DNA technology. So genetic engineering is the discipline that is, so today it is broad discipline, and routinely it is done in practice in every laboratory of the world, and organisms are modified. So due to DNA splicing, DNA recombination, these are recombinant technology, genetic engineering, belonging to genetic engineering. Scientists, biologists in every corner of the world in the laboratory produce modified organism for particular purposes, to synthesize drugs, to modified varieties, and to synthesize another chemical. Just now in 2015, Craig Venter Institute of America, a group created artificial in vitro semi-organisms, means half synthetic and half. So semi-synthetic organism, and total of that bacteria, total